Welcome to Lesson 8 from Unit 1 Validation Science. We're going to be talking about manufacturing processes in this chapter. We're going to be looking at the different types of manufacturing processes and looking at some examples of manufacturing equipment. In the pharmaceutical industry, there are three main manufacturing routes. So an active can be extracted from a natural source, you can have chemical synthesis, or you can use biotechnology. In the organic chemistry, it is dominant in the organic synthesis pharma, so generally your API and a lot of your finished products are using organic chemistry. And we're talking mainly here about small molecules, so in the region of 100 atoms. In the microbiology, which is dominant in the biopharma sector, here you're talking about macromolecules, and you're typically talking about 10,000 atoms folded up correctly. Just to give you an idea here, we're talking about two examples of an organic synthesized product. So the first one is aspirin. Again, we have a small molecule here with a molecular weight of about 180, and you can see what it looks like. And the second one is a small molecule Lipitor. Lipitor is a cholesterol reducing drug, and it's got a molecular weight of 1200. And again, you can see what the structure of it looks like here. In contrast to that, then, you've got an example from the biopharma industry where you've got a large molecule, which is a protein, and it's got a molecular weight of 70,000. So it's a completely different range. So you'd often hear about small molecule, big molecule when they're talking in terms of the different types of the industry. So there are four main types of sectors in the pharmaceutical industry. We've got API, which is your active pharmaceutical ingredient. These are where the ingredients are either extracted or there's chemical synthesis to produce the active part of the product that does the actual medicinal work. We've got the second sector, which is finished product. So you're using API and other excipients to produce your finished product. And then you've got biopharmaceutical, where something is biosynthesized, and then the medical device, where something is manufactured. So API standing for active pharmaceutical ingredient. The active ingredient in the medicines, the bit that actually does the work. For example, the paracetamol and Panadol is your active ingredient. The ibuprofen in your Nurofen is your active ingredient. They're also known as bulk pharmaceuticals or drug substance. In finished product, you have a formulated product. So you've taken your API and manufactured it into an actual product to be released onto the market. And here we're talking about things like tablets, syrups, injections, nasal sprays, and inhalers. Part of the formulation then would include your fill and finish, and we would also expect to see packing operations as part of that process. They're also known as secondary pharmaceutical drug products. In the biopharmaceutical industry, you've got medicines here that are manufactured using biotechnology. They're very often proteins, and you're talking about the different sections of the process. So you'll be talking about upstream, downstream. You would have the formulation of the drug, and then the fill and finish stages, depending on what finished product is going to be like. In the medical device, you're talking about any device intended to be used for medical purposes, and these range from a huge variety of items. So you're talking about simple low-risk devices such as a tongue depressor that the GP might use to check your tonsils, up to very complex high-risk devices that are implanted and sustain life, things like pacemakers. Some of the newer medical devices also would have drugs eluting from them as well, some of the stents and different things, so that they're getting right to the site where they're medically needed. That adds an extra level of complexity to medical device, and we're seeing a bit more crossover. This is just an interesting slide. It gives you some differences between the organic synthesized product and the biopharma industry. So generally you're talking about an organic solvent-based manufacturing process. In the biopharma, it's generally water-based. In API and in finished product, often your aseptic manufacture would only be at the end. The rest of the process wouldn't be aseptic. Whereas in biopharma, the aseptic manufacture is critical throughout the entire process. In the chemical side of things, you have greater flexibility of your equipment and the maintenance of it. In biopharma, it's much more restrictive for your equipment and how you use it. In the chemistry, the product and the process is much easier to analyze. There's set tests, you know exactly what's happening inside in the reacting vessel. Whereas in biopharma, often you're running blind. You have to wait until you see what comes out at the end. So validation is really vital in this industry. In the chemistry, there's different ways to manufacture a product, so you have some options as to how you achieve your isolation of your chemical molecule. In biopharma, you've got a harsh process, so that can actually damage the product, so it's more sensitive. In chemistry, campaign flexibility and changeover and implant process optimization is much easier and is much more manageable. It's a lot more restrictive in the biochemical industry. Unit operations then. So manufacturing processes are made up of a series of unit operations. The majority of processes in a traditional pharmaceutical and biopharma plant, they're physical rather than chemical or biological. And these processes are common throughout the industry, and they're known as the unit operations. And they might be things like mixing, filtration, purification, distillation, etc. This is just an example of a typical process flow from an API manufacturer site. So you've got your raw material going in, you're doing some kind of a chemical synthesis or a reaction here, or perhaps an extraction. You might distill out your product, you might need to crystallize it, you might need to perform some centrifuge or filtration, and you might need to dry it at the end. You may even need to mill it. And once you've gone through all of these different unit operations, depending on what your product is, you will have your API, your active pharmaceutical ingredient, coming out at the end. An example of an API manufacturer is aspirin. And you can see here from the process flow diagram. So you have your acetic anhydride going in with your toluene into your mixing tank, into your reaction chamber. Then you've got your salicylic acid, you've got your cooling tank, and you've got your filtration here. So you're taking out your acetylsalicylic acid. And you can see the chemistry of the reaction here. This is an example of a finished product manufacturing process flow. So you're going to start off here with your API and your excipient ingredients. Just to give you an example, what is an excipient? An excipient is an inactive ingredient. Your excipients can perform a number of different functions inside in the manufacturing process. So you might just need them to be a diluent. So you want to increase the bulk of a tablet if your active is quite concentrated. You might use starch or lactose would be typical things you would use for that. You might need a lubricant, so to prevent your powder from sticking and to help the flow of the product. Magnesium stearate is often used as these. It would be a good example of it. You might need a diluent, again, a similar thing to help the powder flow through the process. Talc or starch can be used for that. You might need a binder to help the particles to come together to help the content uniformity of the product. You might use a disintegrant. A disintegrant is something that when it gets wet, it'll help the tablet to break up. So that helps to decide what part of the body you want the tablet to break up in and where you want it to be absorbed. Things like starch and cellulose perform that function. For certain tablets or products, you might need to add a colour, and that can be for aesthetic reasons, or it can also be for identification. If you had two products that were different strengths that looked the same, you might want to have a colour so they were easily identifiable. Flavours can sometimes be used as well. That's often, particularly in pediatric medicines, to mask the taste if there's a particularly strong taste of your active ingredient or some of your excipients. And again, sweeteners for the same reason. It can be aesthetic or for identification purposes too. So your API and your excipients are going into your mix, and you would have a number of different operations that could be occurring here. You could be blending the product. Blending helps to include particle size distribution. It helps you to get your particle shapes right. Sometimes milling would be used in this function as well, and again that is to increase the homogeneity of your batch and to help the dosage uniformity. Then also another type of processing that might happen here in your mixing is granulation. Granulation is sort of the opposite really of milling. So you're putting globules together. So you can either do this wet granulation or dry granulation. And the reasons that batches would be granulated is to help the flow characteristics of the powder and to improve the compaction properties for tablet formation as well. Once your tablet is mixed, or your ingredients are mixed together, and whatever needs to be done with them, they need to be pressed or put into tablets, capsules, ointments, sterile liquids, creams, etc. So that's your filling process, depending on what form your finished product is taking. This gives you your finished product. It'll then go to the packaging. Packaging is really an important step in the process because the packaging is the traceability of the batch. It gives the identity, it gives the strength, and it gives the traceability for the batch and where it's come from. The medicine goes then to the distribution centres and ultimately out to the pharmacy and to the end users. Biopharmaceutical process flow, I'm going to give you another more detailed example in a minute, but just on a high level, you've got your formulation, your sterile filtration, then it goes to aseptic fill. Depending on whether your product is going to be liquid or freeze dry, it'll be processed here in this section, and then you'll end up with your biopharmaceutical product. So to show you that in a little bit more detail, in biopharmaceuticals, you often hear people talking about upstream and downstream. Upstream is the process where it's defined from the early cell isolation and cultivation to the part where you will harvest your cells. And it's the part really where your microbes or your cells are grown. Step 2 then of the upstream is the purification. So you want to get your desired density of microbes before you would harvest them. And that's done with ultrafiltration and chromatographs. The downstream side then, the purpose of the downstream really is to meet your purity and your quality requirements. And it's usually divided in 3 main steps. So you have cell disruption, if it needs to be come out of the cell. You've got your purification and then your polishing. And then at the very bottom here, you've got your sterile fill. Here's an example of a mammalian antibody production, and you've got a cell culture. This is the upstream part. So this is an example of what an aseptic filling process might look like. So generally from biopharmas, you can end up with a liquid, or you can have a freeze-dried version, which is down here, and you can see. So that's 2 different types of products would result from the process. And the process obviously would be different depending on which type of finished product you were looking for. So biopharmaceutical is really expensive, and you're using mammalian cells in a lot of cases. 2 to 4 million US dollars per year on media materials, 15 to 30 million US dollars per year on manufacturing costs. So that's the material, the labor, and the overheads. 40 to 60 million US dollars to construct and validate a facility. That's just taking an example of a 25,000 square foot facility. The clinical trials require 4 to 5 years, and they generally cost somewhere between 60 and 100 million US dollars. So you can see why biopharmaceuticals are so expensive. Just to give you an example of the selling price of the biotech drugs, so the selling price per gram of the recombinant proteins, so $375 for human insulin, $23,000 for TPA, 35,000 for human growth hormone, right up to 840,000 for EPO. And EPO we'd probably be most familiar with as one of the drugs that are used by cyclists. Particularly, we would have heard about Lance Armstrong in relation to EPO. So it's just to give you an idea of how the price of biotech drugs are so huge in comparison to your chemical drugs. This is just to give you an idea about what a large-scale vessel looks like in a manufacturing site. These are different types of pumps you might find in a pharmaceutical site. The first one here is a diaphragm pump. The second one is a centrifugal pump, and then we've got a peristaltic pump. So that's what they might look like on the site floor. The distillation columns on a manufacturing site are somewhat different to what you might be used to seeing in the labs. So on a much, much bigger scale, you can see the size of this one here. Filters, you go from something very small to right up to something like the size here. Here's an example of a heat exchanger. So you know from the lab experiments what your heat exchanger would look like in the laboratories. This is what one might look like out on the plant floor, and you can see in here the internal workings of it. So back over to you. Let's check your understanding. What are the different types of manufacturing processes? Can you list 3 unit operations in a bio or pharma manufacturing operation? Thank you for your time and attention. We'll see you for the next lesson.